This is a summary of another project that we did with my favorite collaborator, Jana Gevertz. And the title of this project is Minimally Sufficient Experimental Design Using Identifiability Analysis. Okay, so my main job is I do first-in-human dose projections. And what actually that entails is I analyze preclinical data that my teams give me. I describe it using mathematical models. I scale it to humans. I do some magic, and then, ta-da, I come up with an efficacious dose that I can recommend to take into clinical trials. Mostly I work on anti-cancer drugs, and the typical data that I have would be PK, so drug concentration over time, and maybe target occupancy in the plasma, and then some resulting tumor growth inhibition. The main assumption that we have for a lot of this work, this type of work, is that we need to get some kind of sufficient drug to the tumor to get some sufficient target engagement, and we expect that sufficient target engagement to lead to efficacy. We don't usually have actual drug concentrations or target occupancy in the tumor microenvironment, only in the plasma. And we know for a fact that drug concentration and target occupancy in the plasma would be different from that in the tumor microenvironment, but how different is unclear. And so the main questions of interest that I would have here is, can we trust model projections of these dynamics in the tumor microenvironment without actually taking measurements there? And if the measurements in the target occupancy in the TME are needed, then how many samples do we need and when should we collect them? So this is the premise here, is that these are the data that I have available usually when I'm doing my job, when I'm making my projections. If I have other data, then that would expand it. I would find other approaches. But in this particular case, if these are the data that I have in the plasma, can I trust the model? Like, I can calculate plasma concentrations and target occupancy in the tumor microenvironment, but without actually taking those measurements, can I trust my calculations? And if not, if I do need to benchmark it against data, what data do I need and how much of it? So in order to answer this question, we modified some standard PK target occupancy TGI, so tumor growth inhibition model. And what we have going on here, so it would be some kind of a standard structure, central compartment, also kind of a plasma compartment, and you're giving it the drug and it gets absorbed, and maybe it goes back and forth into peripheral tissues, and maybe it goes out some rate, and it distributes into the tumor. And so then hopefully it will find some cells that are expressing the target, will combine with it or bind to the target, initiate some signaling, and that will translate into the downstream effect in the tumor hopefully dying, based on as a function of target occupancy in the tumor microenvironment. So that is really the main premise of how a lot of these drugs work. The drug is binding some target on the tumor. You need to have enough of this to initiate this hopefully self-destruct cascade. The added modifications compared to what the standard model, because this is a very standard model structure that we have, but what we added here is we assume the drug distribution into the tumor is a function of tumor volume. So if you have a larger tumor, maybe you will have the greater absolute drug in there. And if you have a smaller tumor volume, then of course you would have less drug going in there. So maybe you'd have less target occupancy in there because you just physically have a smaller volume to work with. Another thing that we took into account here is that target synthesis in the tumor microenvironment is a function of tumor volume. So again, we're not assuming that there is just some static rate of target synthesis in the tumor, because we assume that it has some kind of a turnover. That is a standard assumption, but we made it a function of tumor volume. Again, larger tumor will probably mean larger target. Smaller tumor, smaller target concentration. And this piece also is an important modification here, that apparent drug target affinity in the tumor microenvironment could be different from plasma. So the way that these things work is your drug is binding to the target at some rate, and then so that would be the K on, and then it dissociates at some rate. And we are assuming that in the tumor microenvironment that apparent affinity, so the rate of this complex formation might be a little bit different than in the plasma. Maybe you have a little bit more of the tumor to encounter. Maybe you have just different distribution of the target compared to how you would see it in other tissues. Really, this particular assumption came from just working with real data and finding that this is what needed to be assumed in order to actually describe the data that we had. So we built it into this model. The way that this was parametrized is we took some data for pembrolizumab, which is anti-PD-1 drug. Keytruda is the brand name. And the paper was from Lindauer et al. From 2017. And what they had there is they had drug PK over time, so the concentration over time. These were, I guess, 3 doses that they gave for 1 and 10 mg per kg. And this was really the critical piece of information. They had the plasma concentration, so these dots that were actually data, compared to target occupancy in the tumor. So the reason we selected these data is because we have this data for this relationship between plasma PK and target occupancy in the tumor microenvironment. Usually we do not have the target occupancy in the TME. We usually do not have these relationships. And the model that we use this model to ask the following questions. First, would we have needed all of these data to correctly predict target occupancy in the TME? Or would a few of these points be sufficient? And if you need only a few of these points, then how much of this data would have been sufficient to uniquely parameterize the model? Okay, so in order to actually do that, we need to introduce a particular method. I promise it's just one mathy slide. And it's called the profile likelihood method. The idea is as follows. You select a parameter in your system that you cannot estimate experimentally, but that could significantly affect your predictions. So this is my system, and in this particular case, we took the target synthesis in the TME as a function of tumor volume, and also this apparent drug target affinity in the TME. We cannot really measure it, but we know we can do sensitivity analysis, we can draw on experience, but we know that these are the things that are going to be very important. Okay, then we're going to determine a range for the values of this parameter. Let's say this is just an illustration, and we'll fix the value of the parameter with some value selected from the range. And for this fixed value, we will find a parameter set that minimizes the standard cost function. I'm not going to go through this. This is a very standard representation of it. So you have the mean, you have the variance, and basically take the difference, square, normalize by the variance. And this is a cost function that we're trying to minimize to make sure that our data is as close to our calculation as possible. Okay, then we're going to save this optimal value of the cost function right here. And we're going to repeat this process throughout the entire range that we have looked at. And with any luck, what we're going to see is what looks like this. And we're going to take some kind of a confidence bound. And we are going to be very interested in these points below the confidence bound. So how do you actually interpret these things? If the curve that you got looks like this, we call this practically identifiable. That is, profile likelihood has this parabolic shape and has a very clear minimum. And that would be your optimal and true and unique value of the parameter. So you can trust that this would be the real value of the parameter based on the data that you have. If you have something that looks like this, that will be called structurally non-identifiable. So this profile likelihood is flat, and that means that there's an infinite set of parameters that give equally good fits to data. So here, the fit to the data would be better compared to these. But here, they're all equally the same. So you cannot tell which of these parameter values is actually a better fit. There's this third intermediate option called practically non-identifiable, but structurally identifiable. But you still cannot really use it to find the optimal parameter set. Because yes, it crosses the confidence bound over here, but really not here. So maybe you need to take a wider range of the parameter, but you cannot actually do anything really practical with this from this case. So really, this is the case that we need. Oh, but wait. the only way to use this method is to use real data to be able to check for practical identifiability. But we want to use this method to determine when to sample to generate the data. So we have a bit of this chicken and egg problem. And technically, yes, you do need real data to be able to check for practical identifiability. But this is how we propose to get around this. And this is really the method. Let us assume, and this is a very big assumption, that the model that we have is sufficient to capture the data that you could theoretically collect experimentally. That is, the model is written for the experiment that will be used to generate the data. We will fix the parameters with strong experimental support, like usually the PK parameters are. We have very well developed tools to estimate those in the plasma. We will fix the least sensitive parameters, and we will leave only the most sensitive ones for this analysis. We're going to call this our vector P. Then we are going to generate some simulated data for this variable by sampling all parameters in the P from a distribution with a specified mean and standard deviation. So what does it actually mean? We're going to assume that my value of the parameter falls within some distribution, or both parameters, and then we're going to run this model with these values of the parameters and plot what the data would look like. And then we would keep doing it and come up with this kind of a noisy simulated data. And the idea is that if we trust the model, and we assume that we'll be able to get these data from the experiment, let us guess what they will be, and then let me describe to you the method, what we're going to do next. In the next step, we are going to check if the parameters in this P are all practically identifiable if all, let's assume, daily data were available. So if so, you can uniquely identify parameters if variable measured every day. Yay. So what we're going to do is say, assume we have all of these data. If we have all of those data, would these parameters be practically identifiable? If yes, wonderful. If not, then we need to fix more parameters until we find the small subset of parameters that are identifiable. So you cannot just take all of the parameters in your system and just go through this. Unfortunately, this was not going to work, but you can find your way to work through small subsets of them. And then we're going to check if identifiability is preserved if you collect the variable of interest that may be only 1 time point, or 2 time points, or 3 time points. So essentially, we're going to take just random time points from our simulated data and ask, if I just had you, would you be enough for me to find the real true value of the parameter? What about if I only had 2 random points? What about 3, etc.? And then that will allow us to design an experiment to collect the data at these time points that we know are identifiable. So the fits that we will get from the model will be unique. And then hopefully we'll be able to trust the extrapolations from it. That's the point of all of this modeling. And this is the idea of this is the model-driven experimental design recommendation that is going to come out from this process. So let us now apply it to our example. This is going to be the series of steps that we're going to take. So let's start with identify an experiment. That's going to be collecting target occupancy in the TME for standard. We're going to take a month-long mouse experiment collecting tumor growth inhibition. We are going to develop, as we showed already, the PK target occupancy TGI model. We have that. We have selected our parameters. So this K-on, dry target binding rate in the TME and rate of drug synthesis in the TME. We have generated simulated data of this percent target occupancy in the TME. And now the big step, we're going to assess identifiability using all simulated data. Okay, so the first question is, are these parameters identifiable if we measure percent target occupancy in the TME, let's say daily? Assume we use all of the data if we have perfect data. Ta-da! Okay, this looks really good. Profile likelihood curves for both parameters have this parabolic shape, and that is below the likelihood threshold of 95% confidence interval. So great, we can confidently identify these parameters if we had percent target occupancy in the 2 microenvironments sampled once a day for 1 month. Glorious. That is an absolutely terrible experimental design and absolutely unfeasible to do for many reasons. So what we want to know is how many data collection points can we peel away and still preserve identifiability. How many of these and when should we take them of our experimental points in order to be able to still preserve this shape and be able to confidently identify the true values of these parameters? Is sampling once enough? Well, let's try. That is a relatively easy thing to check. So we tested all possible ways to measure a single time point of percent target occupancy in the TME over 1 month. In the graph I'm going to show you, sampling from very early time point will be in blue and very late would be in yellow. And this is what we get. Okay. So what we have here, this is if you have all time points. So this is kind of the perfect data case scenario. With K on T, you clearly see that it's not structurally and obviously then not practically identifiable. Pretty much for all one-day samplings, you have this flat profile. There's one case when it is structurally identifiable, but not practically identifiable, so can't really do anything here. So for this guy, the synthesis rate, appears structurally but not practically identifiable for all one-day samplings, or most one-day samplings, because you see that most of them actually cross this threshold at one point or another. Okay, so does it really matter? I mean, that's really a question of what does it do to the model's predictive power? Well, let's look. So assume that we collected the measurement on day 1. And then we are going to run the simulations and try to fit and see what would be the predicted range of tumor growth curves. Well, what you can see is that you cannot predict even the trend in tumor volume, whether it's going to increase or decrease, even though experimental data, yes, is within bounds, but you cannot even tell if the tumor is going to grow or not. So this is not really a useful fit. If you take this, if you can only do one measurement, to take it on day 15. Okay, so here you have relatively actually small variation in the predictive region, and at least you know that the predicted tumor is going to grow, but well, this does not contain any of your experimental data. So maybe that's not the best thing. If you only can take one measurement and you take it on day 29, for example, just at the very end, okay, well, you can predict the tumor volume, kind of range, or at least the trend in tumor volume, although the range is really, really big. And most data does fall within the predictive bounds, but I would not really, this is not sufficient. This would not really be good enough. Okay, so now let's ask the question, maybe we can actually get something with 2 experimental time points. Now this becomes a combinatorially very large problem, but nevertheless, we tested all of the 435 possible 2-day protocols where percent target occupancy in the tumor microenvironment is collected in such a way that obviously the second time point happens after the first time point, and all of them happen before the 30 days. In the next visual, you'll see that some samplings had very small spacing between the days, and that's going to be the blue, versus large spacing between the T1 and T2. That would be the yellow. Okay, now that is interesting. It appears that both can be practically identifiable for some sampling. So remember, the black one is the ideal case scenario, and these are the profile likelihood curves for the different protocols, different samplings. Okay, that is very cool. That is promising, but it is hard to tell if one sampling makes both parameters practically identifiable. So we're going to take a closer look at that. In this analysis, for all samplings with obviously T1, so first sampling point less happening before the second one, let's visualize a number of identifiable parameters. So if you fix the first sample point and then look at the second one and ask, if this is the first one and then this is the second one, is it going to be identifiable or not? And the key obviously here is that the yellow squares, none are identifiable. Okay, the red ones, one is identifiable, and we have several blue ones. So there do exist protocols where even with just 2 time points, you are going to be able to actually confidently estimate those parameters. That's pretty cool. These are the 6 exceptions. So, okay, the first one happens on day 1, second one on day 3, and then on day 3, and the second one would be on day 4. And if you start the first one on day 4, then you actually have these 4 options, day 6, 7, 10, and 13. So that is already very, very exciting and could be potentially quite useful, especially if you have very, very limited resources. The problem with this is it's not robust. There is no room for human error. If you missed, yes, assume you do the day 4 and you ended up sampling it on day 6, but you were not, or day 7, and you ended up not being able to come in on day 7 and you came in on day 8, well, now you lost identifiability. So it's okay. And the predictive regions for these are actually much, much better. I mean, look at this compared to what we had before. It's actually wonderful. Predictive volume is constrained and largely contains experimental data. So we would be able to generally trust extrapolations and predictions from this model. But the 2-point experimental design is not robust. Feasible, but not robust. So maybe we can do better. Okay, so now is identifiability preserved with 3 experimental time points? Now this becomes a ginormous combinatorial problem. So what we did here is we generated all of them, but then we took 1/3 of these possible 3-day protocols just to see if we can get some sense of patterns from that. So non-unique, obviously, samplings were removed, and some samplings had very small spacing between the first collection days. That's going to be, again, the same key. Some had very large spacing. Okay, now let's look at the results. And these look really, really promising. Both parameters are practically identifiable for some 3-day protocols, and looks like quite a few of them. Okay, great. Now let's dive into them. One way to take a look at them, so if you put them on a plane, there were 80 unique, practically identifiable samplings out of over 1,000, and the blue ones are what we care about. And as you see, like if you maybe visually and mentally rotate this, you might see like looks like almost like a plane cutting through this. There is something going on with this blue cluster. So let's take a closer look at that, because it looks like most blue points line along this T1 equals 4. And again, this is specific for this particular protocol that we're simulating. I'm going to touch on that in a moment. And the first 2 points seem to be happening before day 16 over here. Okay, that's interesting. Let's take a closer look. So if the first sampling point is taken on day 4, when should we sample next? Okay, so we're looking at the sampling regions, and let's first look at if the second point is then taken between the days 5 and 16. And that looks pretty interesting. That looks pretty good. It looks like 90% of sampled points in this region result in both parameters being practically identifiable. But it does not guarantee practical identifiability. So let us try to further narrow down the sampling space. In this particular case, so let's assume that we take instead point 1 at day 4, and let's take point 2 maybe between days 5 and 10, and point 3 then between days 16 and 30. And in this case, all of the points that we have here are blue, so all of them are identifiable, practically identifiable. Which means they give one kind of optimal fit, which makes us trust model predictions, if we trust the model. Now we could have a robust sampling recommendation. That would be very exciting. But this was still sparse. If you remember, we only took 1/3 of all the points to look at this. So let's confirm that this is really robust and take all of the 3-day protocols in the sampling region. And ta-da, lo and behold, all of them are beautiful and blue. And so all of these points result in practical identifiability. So this would be a robust experimental design recommendation. That is, if we were working with a team and needed to provide a recommendation for a sampling protocol for the specific experiment, we would say take the first point on day 4, second one between days 5 and 10, and the third one between days 16 and 30. And if you follow this protocol, this design, then when we fit our model, we are going to get practically identifiable values for our parameters. Now, a point about these points in the protocols. In this particular experiment that we are simulating, this is the first dose. This is a protocol that is given, I think, every 3 days for, I think, a week or two. So the t equals 4, that's day 4. That's just after the second dose was administered. So this is not a universal experimental design. This is a question of if we administered a particular protocol, where, as in this particular case, we're giving drug dose every 3 days for however many cycles, then a good time to start your first data collection is just after you finish the first dose and just give the second one. But again, this is the kind of to show and explain the experimental design and the approach to how such an experimental design could be obtained, really for any protocol. And also kind of nice to see that drawing conclusions from initially, at least starting random sampling of space is sound. We took a third, and that was already good for us to be able to identify this sampling recommendation. Okay, so to summarize, we can use modeling to help inform efficient experiment design. You do not need to collect 30 points in order to trust your predictions. You can choose strategic points, but you need to be able to do this. This is not an intuition kind of a thing. This is something that can be calculated. It does require starting with understanding of a feasible and meaningful experiment and also existing constraints, like, for example, duration or number of points that can be collected, etc. In this particular case, we took a relatively standard mouse experiment, and we took a very specific protocol design, which was the one that was used in the data with which we were working. You do need to do a lot of prep work, like you need to estimate the maximum number of parameters a priori. But this is something that was really exciting for me, is that you can estimate parameters here that you cannot identify otherwise. So you cannot really get a sense of what is the target synthesis rate in the tumor in vivo, design a targeted experiment to actually directly measure it. But using something like this, using this approach, you can actually get an estimate of a parameter that for which you cannot design a specific, unique, target-specific experiment, parameter-specific experiment. And I found that to be extremely, extremely exciting, and also very empowering to really try to dive into and estimate parameters that, well, there's no way to measure. You, of course, have to plan for impact of noise and variability, so sufficient sample size, sufficient collections per sample, etc. And obviously, more noise and variability will require more sampling points. We did some analysis on that in the paper, which you are obviously very welcome to look at. And I guess a more of a philosophical point for me was that this confirmed is there is no right model. There is a model for the data, and there's data for the model. And the model here will be helpful if it is designed to inform a very specific experiment. And that, to me, I find to be very important. We have modeling wars about who has got what kind of detail do you have to take into account, what is necessary, what it can actually be trusted. And to me, this project really showcases that it really has to be specific for your experiment if you want to have extrapolatable information. Because if you don't have identifiable parameters, well, you can get, I don't know how you can trust really the extrapolations very much. And that to me is very, very exciting. And also the structure of the model. I mean, here, even for the same question, can conceivably be different, but it can be equally useful depending on the data available to identify and parametrize it. And I find that to be a very powerful piece of information. Okay, finishing up. So this was the paper. It is out in NPJ Systems Biology. This was done when Jana Gevertz was doing her sabbatical in my group at EMD Serono. This was supported, obviously, by EMD Serono. Thank you. And also this NSF grant. And that is what I got on this. Thank you so much.